Well, welcome everybody to our second 2023 Science on Saturday talk. For all of you regulars, I know what you're thinking. Thank you. For you regulars, I know what you're thinking. What did Andrew do to his hair today? No. My name's Arturo Dominguez. I'm the head of the Science Education Department. I work with Andrew. Andrew is out this week, but I have the honor of hosting our second in-person Science on Saturday. Welcome back, everybody. As most of you know, we've been doing this fully virtually for the last 3 years, and we're really excited to be back. We had last week a phenomenal talk, and I'm really excited about this week's talk. It's a hybrid model. We're as you see, we're in person, but we also have at this moment 54 Zoom participants, and we believe we'll go up in the next few minutes. So just a few starting remarks. For in-person participants, masks are optional. We do welcome you to wear them if you're comfortable, or not if you're not. Thanks for your patience with the entry. How was it coming in? Our staff at the booth are fantastic. So this is great. Awesome. Yes. A round of applause. Yes. So yeah, this is the first open meeting since the pandemic. So there will be some hiccups, but I feel we've been doing a good job. And we have a decreased capacity at the auditorium. So overflow participants will be asked to view it in the cafeteria, but I think we've been okay for the last couple of talks. Please hold all your questions until the end of the presentation. Professor vonHoldt will address them, and both me and Bridgett will be hosting the questions at the end. So for Zoom participants, please make sure that your mics are set to mute. Also hold your questions until the end. We'll use the raise hand feature at the end. We also welcome you to put your questions in the chat, but I actually encourage you for Zoom participants to raise your hand at the end, and so that you ask the questions. So we'll do that. Let's do it that way. That we'll take the raise hands at the end. And just so you know, we're recording these talks and they're all going to be posted up on the site. All right. So, where are we? Where's the key? Oh no. There we go. Excellent. Okay, so today's talk will be given by Professor Bridgett vonHoldt. She is an associate professor of ecology and evolutionary biology at Princeton. She's carried out extensive studies on wild and domesticated canines. Her research has significant conservation applications for endangered species like the red wolf and gray wolf of North America. She also has discovered genetic factors that shape the evolution of social behavior of dogs as a result of their domestication from wolves. Everybody join me in welcoming Professor vonHoldt. I really hope this was accurate.
That is, especially because I think I sent it to you, but I still feel weird when you read that. I get so excited, like I want to do that, and I get to do it.
Well, actually, this is a great lead into the question. So,
yeah.
as all of you know, this lecture series was for many years led by our engineer Ron Hatcham. And so in his honor, he started asking these questions that we have continued every year. The first question, what got you excited in joining this field or science in general?
Yes, so as we get lots of personal statements for our grad program application every year, they start off saying, I love biology, I love animals. And I'm no different. And growing up, I lived in Florida as a young kid, and I was just fascinated with the ocean and things that lived in it. Now, I realize I study nothing in the water or anything about it, but that really encouraged me to think about studying animals and ways in which I can help them. So that made me focus on conservation and other sorts of like human interface. How can we teach people on wildlife and sort of the coexistence framework? And as I went through high school, I loved biology class. I got to help out. Our biology professor had everything under the sun living in tanks and cages in the biology classroom. And it was just amazing. So I think that was the start of it all. And, I kind of weaved my way through various activities and volunteers and just, I was everywhere.
That sounds awesome. And I think you might have touched upon the second question. Were there any teachers that inspired you when you were young?
Yeah, my high school biology teacher, the one with all of the living great things in the room, was one of the first people that he would tell his class constantly, when you're a professor of biology, you will be teaching this. And he was just always talking about, you're going to be doing this. And that was really, it has made a long impact on me. And I think about this a lot. And as I then continued to find ways to interact, there were all of these people peppered across my history that would tell me, oh yeah, of course you can do this. I never came from a family that had a lot of higher educational experience. So they taught me about where I can get scholarships and funds. Because I really thought at one point, like, I don't have money. I can't just pay this tuition. And people were helpful along the way. And that, I think, made all the difference for me in pursuing not only, college was never a question for my family either. Both my parents lack a higher educational degree. My dad was in the Air Force, but had a vocational degree. So it was really great to see my parents support going to college. But then after that, it was a mystery. And these people popped into my life telling me where I could apply for money, or that they're going to pay you when you want to look for your PhD. And that was, I never knew that. So I think there's a lot of people who have said and shown things to me that helped me get here. Of course, I didn't do this on my own.
That's such a great story there. And the last question, and by the way, it relates a lot to, we were talking about this earlier, about bringing folks into the field and how varied fields they come in and backgrounds they come in, and we have to adapt to that. So that's fantastic.
Yeah.
Then the last question, when you're not doing this work, although this work sounds like what people like, interacting with animals just sounds amazing. But when you're not doing this work, what do you do on your free time?
Yeah, so my really nerdy answer is, this is my hobby in the free time. I love when I'm not in meetings and I get to be at home on a Saturday or Sunday, and I've got a full 8-hour workday that I get to sit and do my work. I get to write, I get to do analyses. I love that. But I also have a dog and a husband, so I have to entertain them. So we go on hikes, make sure the dog gets out and about. And then the picture that you had of me, one of the hobbies, which I don't get to do all the time, is go out and look at wildlife. So when I get to visit Yellowstone National Park, which is one of my study sites, I really enjoy seeing the animals. I work with their DNA, so it's a clear liquid in a tube, and I've got thousands of these tubes, and it's really nice to remind myself and my students that these are animals, and they move on a landscape, and they have lives, and they have drama, and we get to see this. So I love going to Yellowstone. I also work in Texas, where they have lovely coyotes and red wolves there, so I get to see them, and that's amazing. I can't visit enough. And the 1 or 2 times a year I get to go out in the field is just amazing for me.
It sounds fantastic. And
that's my hobby.
We're very, very much looking forward to your talk. Let's welcome again, Professor vonHoldt.
Thank you. Okay, I have to do this. So I'm going to share, and that will override yours. Or maybe you weren't even sharing. Okay, good. That's good. Everyone sees that. Perfect.
Good. Why don't
Let me make sure my, oops.
Let's change the view up there.
Oh, I see. To take that. Yeah, you're going to have to do that for me.
Am I doing this? Yeah. Okay, I don't,
I think there is a
I think
way.
I think we're good though. It's not blocking off the image, so that's. should be
It's
fine.
not. If we come to a point where it's challenging, I'll do something.
Okay.
I'll wave my arms.
Thank you. Yeah.
Great. So I'm so excited to talk about this different part of my research. This is not for wild canine sort of questions, but the other end of the questions I like to ask have to do about evolutionary change. And what better species to think about how we as humans have changed a species over time is that of the domestic dog. And I also love this because you get to see my dog. So this is my baby girl. Her name is Marla. You will see her at least one other time in this talk. And the title of this talk, Everyone is Your Friend. I'm going to give you my history of exploring and discovering some of the molecules that shape hypersocial behavior in canines. This is astounding, mostly because when we think about where dogs came from, we know that their closest relative is a gray wolf. So this is a picture, actually, I love this story. This is a black gray wolf in Alaska. He is known by the name of Romeo. Romeo is no longer in existence because Romeo loved to interact with dogs. So this is a real picture. I didn't Photoshop this. And Romeo was notorious for playing and socializing with domestic dogs. And this eventually, as you might imagine, could cause some concern about the safety of everybody involved. And I believe Romeo was poached at one point. I know, I understand. I've been trying to get his DNA, but this is very challenging. So the story that I meant to tell is that we have this beautiful domestic dog standing next to its wild ancestor, a gray wolf. And humans have been the primary person, the primary driver in changing this gray wolf into a dog. And the change from a wolf to a dog took place over many thousands of years. And we've changed so much about the wolf. We can imagine all of the diversity we see in domestic dogs. In fact, what we can tell you is that dogs have been very good at us understanding the genes that have changed from being a wolf gene into a dog gene that's associated with traits. So as a geneticist, I'm always interested in looking at the mutations that change the way something looks, behaves, sounds. Is it fuzzy? Is it wiry? Anything about it. And dogs have proven to be a really good model for this. And in fact, over the years, we have known and discovered many of the mutations that connect to the way a dog looks, the phenotypic diversity. So as a reminder, we have these two terms that I will probably be talking about, pepper them in. Genotype is the collection of our genetic makeup, and the phenotype is the presentation of all of those genes acting together in the environment that the animal is living in to create the way it looks, it sounds, maybe its size and shape and texture. That's our phenotype. So for dogs, we know that they have this huge diversity of phenotypes. And when we start looking at the genetics between them, we're looking for a gene that might tell us about the way it looks or behaves or sounds. So I'm going to run through a couple examples of where we have discovered genetic mutations that are connecting to a trait. They cause a certain trait. And what's surprising is that in domestic dogs, we've run this experiment. We've already artificially selected and bred dogs extensively for hundreds of years. We have whittled out all of this genetic noise, and we can pinpoint the tiniest fraction of mutations that control traits. So for this example, we see and have found that a single gene is responsible for changing the curly tail to an erect tail. A single gene, that's all you need to change. For droopy ears and prick ears, those that stand up, this is now controlled by two genes. The leg length, so stubby-legged dogs. You can take a giant greyhound with these beautiful long legs and change the action of a single gene. And that will cause the abrupt stop, the growth of the long limbs will stop growing by simple one genetic mutation and result in all of these 4 little stubby legs on an animal. And I think this is just a dramatic example. Finally, for the way like a dog looks, how it feels, and how much fur and where the fur is growing, there are 9 different fur textures and lengths, 9 fur phenotypes controlled by the collective action of 3 genes. So if we have a dog that has both long fur and it's kind of wiry with fur that's growing along the arms or like a mustache, those are called furnishings. So then you would need collectively the action of 2 of those 3 genes to result in that phenotype. So this astounds me because these are really diverse traits, very dramatic changes, and it looks like we only need up to 3 genes acting to create such a dramatic difference between dogs, and also when we think about what wolves look like. So this is great. This is how a dog looks. But now we can think about additionally how a dog might behave. And that is also a feature of dogs that is quite diverse. We know dogs are collectively grouped into sort of these clubs or functions that they perform. So if you have a sporting dog or a retriever dog, those are names that tell us something about the function and the capability of that dog. So here we have a couple examples of retrieving and some herding. These collective behaviors are very distinct, and we could then anticipate that there should be some genetics shaping that. Similarly, for more assistance dogs, these are dogs that can either help people. We have sort of seeing eye dogs in the middle or assistance dogs. There's super sniffer dogs, whether they're super sniffing out chemicals or drugs or other sort of weapons. And then those that are biomedical dogs, dogs that can sniff certain types of cancers or can anticipate changes in their human, maybe panic attacks that are coming to onset for their human. They can anticipate this and provide care. And then there's a slew of other behaviors, some of which I think are just fantastic. Those that are the purse dogs, in my opinion. We've miniaturized them so we can take them with us everywhere. They're in our bags, they're in our backpacks, they're on the subway trains with us. Dogs that perform other sort of intellectual functions and then other societal functions. So we know that we can create dogs that fit very specific roles. And a lot of this, we are always searching for the genes that might control that. And studies have been done less focused on the genetics, but more so about how do we actually create a dog with a very specific function? And how is that process, how quickly does that process happen? And it always turns my attention to this classic experiment that was done on red foxes. So in the mid-1900s, a researcher, Professor Belyaev in Siberia, was obsessed with understanding the domestication process. How do you turn a wolf into a dog? How quickly can you do that? And what is needed in order to see that? So what he did was he established a very experimental breeding program for red foxes. And he chose foxes predominantly because in Siberia at the time, and probably still continues on through many cultures today, that there are farms that raise animals for fur. Fur is a very important part of a lot of weather-based gear and materials and clothing. So all Professor had to do was look around him and was surrounded by fur farms that were raising foxes, wild foxes, breeding them for fur. So he got some fur foxes from a local farm and decided to start breeding them for a very specific purpose. And for him, that was friendliness. He imagined that the way we turned a wolf into a dog was by selecting only the friendliest animals. If we had control in a litter and we could say who could be reproductively successful, who could breed as an adult, we would probably want to limit that to only the pups that grow up and show a fair amount of friendliness towards people. So Professor Belyaev did the same thing. For every litter of baby foxes that were born, they were extensively tested for friendly behavior. These were tests that involved a human experimenter walking up to the cage and writing down over 300 traits that the fox might exhibit in response to the presence of the human. Everything from, are the ears forward or backwards? Are the teeth being exposed? Are they making sounds? Are they chirping sounds or are they growling sounds? Do they approach the edge of the cage where the human is, or do they sort of go back to the corner and hunker down to try to get away? And when those animals that had all of those friendly traits were found at the end of that generation, only those were allowed to breed, and they would produce offspring. And by Darwinian evolution, we expect that those offspring are more likely to have those friendly traits. So he did this generation after generation after generation. So about 45,000 foxes in this experiment, took about 50 years to breed. And in fox time, this is about 32 generations, he went from a fur fox that looked just like that to a fox that looks, in my opinion, not that different from a border collie or like an Aussie or something. So this looks like a dog. It also acts like a dog. It kind of sounds like a dog. It has a lot of whimpering, and it will roll over when you approach it because it wants to interact with you. In fact, so many things about this domesticated fox are indistinguishable from our own domestic dogs. And that was only a 50-year experiment. Granted, this was a very strong selective process. All the animals that didn't fit the bill for being friendly, they did not reproduce. So this is a strong selective experiment, but we have now all of these domestic foxes and show us how they are entirely overlapping with what we imagine a domestic dog is, the way they act. So when I think about domestication, there are lots of traits that have to happen at once for an animal to be called a domesticated species. And we call that collection of traits the domestication syndrome. And the word syndrome only means that there's a collection of traits that always occur together. So for an animal to be considered domesticated, it has to have all of these features co-occur. We see that there is a change in physiology, something like how many reproductive cycles it can actually have per year. Wolves can reproduce once per year, no more than that. Dogs can have up to 3 litters per year. We also see changes in development between wolves and dogs, just even as early as when they open their eyes and their hearing starts to develop is different between wolves and dogs by a matter of a mere 14 days, maybe 18 days. But this is incredibly important for how they explore the world. For dogs, they have all of their senses develop at the same time, whereas wolves have them staggered, and that means an entirely different experience as a baby. Anatomy is different. We know that every skeletal bone and muscle has been changed in domestication. We have tiny things, we have long things, we have tall things, we have stuffy faces that are short and stout. So we know that anatomy is very different. Cognition is quite different between dogs and wolves, and we'll talk a little bit about that today. And I'm mostly focused on behavior. Wolves have collectively a generalized behavior that we can all talk about, and dogs span this entire array, quite different from what you see in wolves. So I want to focus on behavior today and give us a sense of how that's different between wolves and dogs, and kind of search for the genetics behind this. So wolves, I'm showing on the left, and they're generally showing this low tolerance for human presence. They're usually going to avoid people. They don't have a tremendous interest in people. They don't really want to seek out that interaction. I know there are stories that we always hear about throughout literature or maybe some popular news of that friendly wolf. White Fang is one of them. This is a fictional story about a wolf that has a close relationship with a person. And this does happen, but this is a rare occurrence. This is not the stereotype of the species. And I can talk to you about it later, why I think that actually happens. So the low social sort of interaction, I'm going to continually represent as a gray brain throughout my talk. Now, on the other end of it, when we look at dogs, dogs have a completely different interaction with people, and they have a reduced level of fear for people. And this is what we see in the domestic fox experiment, the friendly versus the fearful response. So dogs don't have that fear of people generally, not to the extent that wolves do. And in fact, dogs have this increased desire to play and interact with people with respect relative to that of the gray wolf. So for dogs, or this social brain, we're going to keep it as an orange color. So as we go through, what I set out to do, how long ago did I start? I first started this work in 2006. And so that's many years ago. This was during my PhD, and part of my dissertation, I wanted to understand the genetic changes over evolutionary time between wolves and dogs. And in order to do that, we could set up a very simple case of sequencing as many gray wolf genomes as possible and compare that to as many dog genomes as possible. And I'm simply looking for mutations that are different in the two types of genomes. That wolves might have a collection of alleles, that's what's represented by these dots on these vertical chromosomes. That wolves might have some amount of genetic diversity, but that genetic diversity is not found, generally speaking, in the domestic dog genomes. In fact, what I'm looking for is that all domestic dogs share the same types of mutations, pretty much to the exclusion of gray wolves. And that means those mutations might be responsible for making it a dog. And what we see here, when I went about this search, is that indeed there are a cluster of mutations on the canine chromosome number 6 that are specific to dogs. And not only are they specific to dogs, they fall within a gene. And this is kind of a mouthful of a gene, and I'll explain this gene in a moment. This gene has a role in this syndrome in people called Williams-Beuren syndrome. And those mutations are almost exclusively private or found only in domestic dogs. But some of you might have noticed there's one over here. So we're going to keep that in mind. What we do know is that over time, we have learned that domestic dogs didn't just sort of appear out of nowhere, right? They have this ancestor, and there's a lot of traits in this ancestor that are important to leading us up, evolutionarily speaking, leading us up to be a dog. So there's some ancestral genetic changes that occur at a rare frequency. It's rare for a wolf to have it, but maybe the white fang wolf had something like this. And we needed that in order to establish and make a dog different. So all of these genes that are unique to dogs, all of these mutations that are unique to dogs fall within a gene that is involved in this syndrome, Williams-Beuren syndrome. Now this syndrome, okay, now my slide is frozen. Yeah.
Click presentation.
Okay. Thank you. If it advances, I will quickly back up. Okay, so the syndrome, Williams-Beuren syndrome, is this multi-presentation sort of characteristic syndrome. It has a whole other branch of traits that I'm not going to discuss in more of the clinical and physiological presentation. I only really want to focus on the behavioral aspects of a person with Williams-Beuren syndrome. And when I initially read through the description of the syndrome, it's very difficult not to think about a dog. Some of the main distinguishing and clinical diagnostic features of Williams-Beuren syndrome is having a person with this unusual cheerful demeanor, which is a hypersocial person that lacks, as a child, stranger danger. And as an adult, you might call this social butterfly. This person who is constantly socializing with anybody that will basically respond to them. and they get distracted with socializing. I had a friend in college who I think back, I'm like, he like didn't really want to go to class. He wanted to just like go hang out with everybody. There's also other features of this developmental program and cognitive capabilities that get a little bit disrupted. So some of the developmental stages have different timing onsets compared to people without this syndrome. And as we jump down to this last bullet point, auditory hypersensitivity, I often can imagine how my dog may not love listening to the vacuum cleaner, or like as I pull out a rack in the dishwasher and all of the glasses are clinking. This can be very kind of disruptive to some animals. So the overlap is quite astonishing to me. Oh, there my clicker works. And the human researchers studying Williams syndrome have discovered so much about the causes that lead up to and are responsible for all of these traits. Now, it doesn't mean all of the dots have been connected, but we do know that in order for a human to have Williams syndrome, they have to have this giant mega deletion on one of their chromosome number 7s. So just to remind us all, we're diploid, which means we carry 2 copies of our genome. We inherit one from our mom and one from our dad. So each chromosome is represented as a pair. And this deletion is called hemizygous because only one of our pairs is missing this giant chunk. It's 2 million bases that are missing. And in that 2 million bases, there's about 30 genes that get deleted. And I'm only showing you a handful of their names here. Now, this means that we have far fewer genes available to do the job of this section of the chromosome. And over lots of studies, both of humans and mice, we know that this one gene listed in red is critical to cause Williams syndrome. If you have this gene, it's called elastin. If it is deleted and only has 1 working copy left, instead of other parts of the genome where there are 2 working copies. In the 1 working copy format of elastin, this is when Williams-Bjorn syndrome will absolutely be presented. Now, it's interesting because elastin is a gene that makes proteins, and this is what genes do. And the proteins often have jobs that are very critical. Some proteins can be hormones, some proteins can be enzymes, and in this case, some proteins can be receptors. This protein makes our cells elastic. Elastin makes proteins that help our cells with this elastic kind of flexibility. You can imagine our circulatory or respiratory system needs to have the flexibility of our cells to move, and that's what elastin does. But we still don't quite understand why the deletion of one of these gene copies of elastin results in the hypersocial behavior in people. We don't know this, but we know this genetic makeup of Williams syndrome in people. And this deletion now removes some of the protein products that are produced by those genes that now sit in this region with only 1 copy. So each of these genes in that region, we know produce some amount of protein product. And we know that this is reduced because when we look at a person with the normal 2 copies of those genes, they're producing gobs more of these proteins. So the reduction in that protein level is a very important feature that causes Williams syndrome. And in fact, what people have done is experimentally created this deletion in mice. Since mouse genetics is a very nice model system for humans, we can just manufacture that deletion in mice. And when we do that, we get hypersocial mice. These are mice that love to be social. And if you don't have this deletion in the mouse, you have our typical sort of solitary singleton mouse. So this is really nice to see that we can hone in on experimentally that this deletion does something critically important for behavior. And this leads me now to the title of the talk and connecting it to dogs. So Williams-Bjorn syndrome, it has just been studied for decades, and we know a lot about it, and it has a slogan, and that is, everyone in the world is my friend. And I think this applies to dogs. Who else would let you do this to them? The look on this dog's face is amazing. My cat would not let me do this, but my dog would. I have pictures. Oh, I should have included pictures of my dog wearing hats. I just love doing this to her. And she looks at me with the same look, like, all right, get it over with. I know you're happy. So I think that there must be several features overlapping human Williams-Billings syndrome and our own dog behaviors. And I set out to now look for the genetic differences that might tell me, are there mutations, are there molecules in our social dogs that shape how social they are, and that we really don't see them both in wolves and also some of these dogs that just don't care to be as social as us. So we did this in a way that was very quantitative. In order to link genetics, so I mean, I've got the genetics down. I'm not worried about sequencing and looking for mutations, but we want to say that this mutation does something. So in order to do that, we needed to collect behavioral data, and not just any behavioral data. We wanted to compare and contrast basically the cognition, the capability of an animal with the decisions it makes for being social. And we collected this data on about 8 wolves. These were wolves that are captive wolves, human raised, so they're familiar with people, and about 16 dogs, pet dogs, not even a purebred dog, mixed breed dogs, the regular sort of dog companion we have at home. And we split this behavioral measurement up into 2 categories. And the first one is a solvable task. We wanted to look at the ability of an animal, dog or a wolf, to solve a task. And the task is simply, here's a Tupperware with a summer sausage in it. Get the summer sausage, and you have 2 minutes to do it. So we have an alone condition. We present the animal, a dog or a wolf, with this treat. We measure how long it takes in those 2 minutes, or maybe they don't even do it in the 2 minutes. And then we swap it up. We're like, you think you can get that summer sausage? Now do that in the presence of a human, a person you know, maybe your owner, maybe a friend of an owner. And we measured across these 2 conditions how much time the dog spent looking at the person or the wolf, and then how much time the animal spent actually looking and working with this puzzle box. So we're calling this attentional bias because we've introduced a distractor. Hopefully, it's a distractor. And then we paired that with the sociability task. We wanted to measure by the decision of the animal, when they have an opportunity to get in close proximity with a human, do they decide to do that, and for how long? So we have another 2 minutes section of time chopped up now over 4 treatments. We have an experimenter always present. They're sitting on a chair, and we draw a 1 meter circumference circle around them and consider that the proximity to the person. And we give the dog or a wolf opportunities to approach and stay close to that person in 2 different phases. Whether or not it's passive, that means the experimenter cannot say or do anything. They're looking at the floor from their seat. They're not touching the animal. They're not saying anything. They're just being there. Versus the active phase. After the animal initiates a greeting, then you could respond verbally. And then we wanted to swap out the identity of the person. If Williams syndrome is truly what we see, then we should see something about strangers, that a dog or a wolf may not care if you are familiar or if you're a stranger. They might act the same way towards you. So here's an unfamiliar person in the same passive or active treatments of this. And we could get 2 different measurements from this study design: how hypersocial the animal is, just overall, how much time are they spending in proximity to a human, regardless of their identity. And then we use the identity. Explicitly for the stranger condition, how much time are they spending close to that person? And the results are generally overlapping with what we find before, and then we get to build on that. So here are the bar graphs, but really I like talking through it. So what we found, consistent with all of the previous work, is that wolves do better at this than dogs. Wolves just chow down through the plastic Tupperware. They don't care. They get that sausage in no time. So if dogs aren't solving this task much, we are able to now tell you why. Why aren't they solving this task? Well, they're busy looking at the person. They're just looking at the person. There's sausage in the town. Nope, we want to look at the person. All right. So it doesn't sometimes even matter who the person is. We see that the dogs are spending far more time closer to the human than the wolves are. And what's really, I think, key for the power of our genetic work is that we were finding that there is still variation, that there are some dogs that don't really care about the people. They're a little bit aloof. And yet there are some wolves. In our 8 animals we tested, one of them was very curious about people. So we had this kind of crossover. It wasn't a species-specific difference. We actually see wolves can be friendly and dogs can be aloof. And this means we get to rely solely on the behavioral data, not the species identity, and look for mutations that might tell us about this behavior. I think I froze again. So then here's the genetics part that it's so straightforward. We're just going to sequence the part of the dog genome that contains the Williams-Beuren genes, and then my original favorite gene, that one that was the mouthful. And all of these genes sit on this little part on chromosome 6 in the dogs. So it's only this little segment at the top part of the chromosome. The blue boxes show where the genes are. You'll see here on this right-hand side, this is that big cluster of about 30 genes that gets deleted for human Williams syndrome. There's our elastin gene, it's right there. And then over on the left, this is my favorite gene. And also what caught my attention is that it's sitting next to this autism susceptibility 2 gene. So this entire bit of the chromosome clearly has some involvement in shaping behavior through a genetic mechanism. So we sequenced all of this. This is about 5 million nucleotides. And I searched for major chromosomal differences. I'm not looking for one nucleotide that's different. I'm taking my advice from the Williams syndrome community that we're looking for major macro scale chromosomal abnormalities, maybe big parts that are deleted, or maybe some parts that are newly inserted. Perhaps there are duplications, and sometimes there can be inversions where you flip the way that you read your sequence from head to tail. So I'm looking for this, and what we were able to do is find just under 90 of these types of mutations. And we could test now how this segregates with the behavior. So are the animals that are far more friendly, do they carry more of these mutations or fewer of them? And that will help me hone in on the molecular mechanism behind the social behavior. So statistically, I wanted to ask, are any of these genetic mutations found at a higher frequency in social animals? And here's the graph. We had 3 measurements, but what you'll see up here is not the hypersocial measurement. We have the attentional bias and the social interest in strangers. Those are the 2 measurements that had significant results. And here are our standard, these are called Manhattan plots. We have parts of the chromosome on the x-axis and our significance value on the y. Anything above this line, so anything in the orange box, is a statistical significant finding. And I've now just named the 2 genes and the 4 mutations that caught my eye. These are mutations, major chromosomal changes that happen in genes. They involve a gene, which is a functional unit. And what we see is that when there's a mutation, again, my favorite gene popped up. I was thrilled. And then I have another new candidate to think about. The mutations in these genes dramatically change behavior. A single mutation in my favorite gene will change the attention of an animal by over 50% of what the average population will show. The same is true for this other new gene. That same impact, dramatically increase that attentional bias. And then finally, Gesundheit, we have this other gene for the hypersocial towards strangers. That one mutation in that gene essentially means you can go from having no interest to a totally obsessed animal with a stranger. So a dramatic effect. And when we look at these genes, so I found the genes that carry these mutations, and I wanted to know what these mutations actually are. And for me, this was an amazing moment in my life because I love transposable elements. If you're not familiar with transposable elements, these are little bits of DNA of a chromosome, like this little orange fragment here. This can copy and paste itself, just like in a Word document, copy, paste, copy, paste, copy, paste. That's what this little fragment of DNA can do. It has the machinery to copy itself and then jump to another location and voila, have a brand new duplicated copy. They used to be called viral DNA. We now know better. They're not just viral. They can perform very important functions. They can disrupt genes. They can create new sort of genetic combinations. They're really important for evolution. But this kind of transposon, this copy and paste, means that we should see a lot of variability. And when I look closer, we get a better sense of how they function. So to go back to the importance, these transposons, these bit of jumping copy-paste genes, can happen so rapidly. In fact, every time there's a new generation, so new offspring are born, that is a probability. That's an event where new transposons can insert new places in the genome. So we can have a rapid mechanism for evolutionary change. In humans, 1 out of every about 100 human births has a new transposon inserted somewhere. This is a very high mutation rate. In dog genetic studies, we already have found that a lot of phenotypes, 3 phenotypes here, are due to the new insertion of a transposable element. We've already seen these 2 examples, the stubby-legged phenotype. This is a transposon that changes how a gene is expressed. It shuts off the long bone growth. We also know that for dwarfism to go from this giant Great Dane into this little teacup Chihuahua, you just need 1 transposon that inserts and again changes the way that growth factors, these proteins that promote growth, overall, how that changes due to 1 transposon. And finally, there's the Sheltie example. For those of us who know about Shelties well, you have several color variations of them, sort of this normal coloration, and then we have this 1 dilution color that is due to the insertion of 1 transposon. And if you get 2 insertions of that transposon, you have an animal that is this deeply merle pigmentation. This is like a very dilute color variant of the Sheltie. But this is a variant that also comes with a lot of auditory and vision abnormalities. Usually they're blind and deaf, or some combination of those. So we see the actions of transposons, and now I can start connecting them to our social behavior. So if I go back to this little experimental design, and I want to show you the real data underlying it, we have these 4 treatments. When I look at now at the x-axis, the number of mutations, this is my favorite gene. We have up to about 4 insertions, 4 mutations an animal could carry. The y-axis tells us how much time that dog is spending next to the person. And we see that it kind of doesn't matter in the unfamiliar or the familiar case, if the person is not saying anything, the dog doesn't care, the wolf doesn't care. There's no real difference that the genotype is making. When the person is responding to the animal, it matters what their genetics are. The more mutations that animal has, if the human responds, then that animal is on that social interaction so fast, they stay at least 3 times longer next to the person if they carry this genetic makeup. So this was amazing to me. And what we're now doing, I'm just going to wrap this up with more of a deep molecular dive. So you're going to learn about some proteins here, I hope, is that we're starting to think about what these transposons are doing. It's one thing to associate with the behavior, but I want to know how does this change social behavior? So we normally like to think about chromosomes in these linear fashions, right? This is that linear drawing. And really what it is a bunch of DNA that's wound up really tight, congealed. And when you string it out, that's the way our genes can act. That's when expression happens. So really, we have this spaghetti string, and even when we unravel it, it's very important that it can still kind of bend and loop and interact with other parts of the chromosome. So you can imagine all of these little parts can come into contact with each other. And this becomes very important when we think about how genes interact with other genes. So the transposon insertions actually do a job. They regulate how genes interact. When we look at what a wolf has, or a non-hypersocial canine, our little gray brain, we see that there's actually this insertion in the ancestral part. The wolf will have this insertion of my transposon in my favorite gene, and only when the insertion is present will my favorite gene make its protein product. And it does that through an interaction with this gene partner. It's stabilized through some other protein action. So this is how you get a non-hypersocial canine. This transposon insertion allows for this interaction. It makes some protein that does something, but it's not involved in social behavior. When we look at dogs, what we see is that the dog lacks this transposon. This transposon no longer can facilitate this interaction. We now don't have the loop on this chromosome. It's now a linear bit of DNA. It has no interactions. My favorite gene can no longer express. And in fact, it's because the gene partner is making its own protein now. And the protein it makes deliberately shuts off my favorite gene. And this is the only way you get a hypersocial canine. So this is above and beyond what I ever imagined we would be learning. And my grad student is still getting all of these bits and pieces filled in. So one day we won't make it all the way to how does this shape behavior, but we're working to see how these gene interactions lead us to think more about the ecosystem of our DNA and not just a single gene doing something. So this is now an incredible convergence and connection between a human syndrome and dog behavior. We have the same genes disrupted, but they're disrupted in different ways, and they produce the same outcome, the same hypersocial behavior. Humans have it through this deletion. Dogs seem to be having this as a result of the insertion and then the lack thereof. So what we're getting is this story that has the same phenotype with people with Williams syndrome and dogs that have this same internal driving desire to interact with people. And what's amazing is that I separately also work with assistance dogs, and this is an assistance dog company that produces dogs for helping people. And the Williams-Bjorn Syndrome Society has, I've connected with them, and this has been a really tremendous moment for me. I think watching how we as humans know how important dogs are, but we also have to remember how important we are for the dogs in that social environment. And Williams syndrome plays such an important role in that, in reminding me how important these interactions are and how this has really shaped our evolutionary history. Dogs certainly wouldn't be here if it weren't for us. And now I sometimes wonder, would we be here without having domesticated dogs? So, the one last slide I wanted to show you. Such a lovely part of my career that I never imagined is that I got interviewed by Anderson Cooper on 60 Minutes. And I was, again, just like starstruck. I couldn't believe that someone would be interested in talking to me like that. But more importantly, when I watched the episode, my dog Marla got so much screen time because they explicitly wanted to see my dog. And I got the screenshot. I love it because it's the point in which I'm talking about. They're saying, I discovered the friendly mutations. But the part of the interview that got quoted, like the public clickbait, was this. Because Anderson asked me basically, does my dog just love me because she's got the genes for it. And like, is this a real love, or is this like a prescribed love? And then my quote back was, nope, even though she has a genetic predisposition to wholeheartedly love you, this is more than she could probably handle. So I loved that. And if you can just Google it if you haven't watched already, but you'll see my puppy. So I want to thank you. I'm excited about what questions you might have for me. I'm very excited to be here, and I hope you've learned something a little bit about proteins, if not a lot about dogs.
Thank you. Thank you so much, Professor vonHoldt. This has been really great. I just kept thinking of my dog throughout the whole
Yeah.
presentation. So we have time for a bunch of questions. So is Britt, where is Britt? Hold on, our mic runner is coming. There she is. She's coming with mics. So yeah, we have some questions, and I encourage participants online to also raise hands and ask questions.
Yeah, I'll try to keep
There
it.
are a couple of questions here.
Yes.
Well, the mics are coming. Just a sec. Okay, one here and the next one here.
And I see it. There's a chat over there.
Go ahead.
So I had a question. So would you think that as the population of humans is becoming larger and more densely populated, does that encourage more social mutations like Williams-Burns syndrome? And if it does, would you say that it is making us less task-oriented like it did the dogs?
Oh, so for the people side of this?
Yes.
I'm not a human geneticist, but what I can talk about is the probability. So as you increase human population, you have many more chromosomes being produced. And with more chromosomes being produced is always the opportunity that a mutation can happen. Yes. So purely based on numbers, I would anticipate that, well, also our technology is getting really good at kind of detecting very nuanced phenotypes, new disorders, new syndromes, new diseases, new conditions are being described. And whether that's because technology is better or because there are more chromosomes in which something can happen, I'm sure it's a combination of both. Yes, I'm not sure about that distraction. I like, as an older person, to blame technology on that. But absolutely, I think as genes have important functions in attention and several other complex traits, it may not be a bad prediction.
So we'll take one online. Robert Krovitz, can you unmute? Anthony, can they unmute on the Zoom online? Okay.
It looks like yes. Yeah.
Robert, can you unmute? Looks like they, oh, there we go. Yes. Robert, can you speak? I'm not hearing anything, Anthony. Do you think they need to unmute on the computer? Okay. In the meantime, I think he wrote out his question, but I do want to maintain folks online to please raise their hands. Robert was asking, is there any connection between these genes and oxytocin?
Oxytocin.
Oxytocin.
So oxytocin being a hormone that helps with the bonding experience. We currently are looking at a whole different study of dogs that do have various hormone assays. So oxytocin is one of those that helps promote a strong social bond. This gene, well, the mutations I've looked at, the genes don't have a central role in oxytocin, but these genes function very early in development, which means it sets up a whole path kind of that it doesn't deviate from. So if that full path results in changes in oxytocin, that's several degrees separated from the mutations I'm looking at, but certainly bringing those kinds of hormones in really help. What we're trying to do now is look at sort of that question, that bonding question, but for puppies in their first 6 months of life, depending on how they're raised by their mother or how they're raised predominantly by people. How do these mutations maybe shape the behavior at that early life experience where oxytocin is very important? And can people elicit that response in the pups as well as their own biological mothers can? So it's complicated, but yeah, I think we think about that a lot.
Excellent.
I hope that helped.
Hi, that was a fabulous talk.
Who's got the mic? Oh, there. I was like, I saw that one there. Thank you.
As the father of a dyslexic, I finally understood my whole family based on. But,
Okay, good. I'm glad I helped.
so, but I'm always a little worried about genetic disruption. Yesterday, I met with some guys who, you know, that the apolipoprotein is responsible for Alzheimer's syndrome. And these guys are from Utah, and they apparently the state of Utah has integrated its health records
Yeah.
with the
Right.
with
like
the
their
genetic,
Mhm.
genetic Mormon history.
Yeah.
And what they found was there was a set of people who actually had the Alzheimer's gene who didn't get Alzheimer's, and it was apparently connected to another protein. So I'm wondering about how these protein interactions, you know, it's clear that there are other genes that influence the genes that happen. So do you see that kind of thing?
Yes. So a lot of what's under the hood that I didn't show you today is us looking at what are the proteins being made by these genes? Where do they go and interact? Are they interacting with other genes, or do they interact with other proteins? So we're trying to build up that modular network, which will help us know who are all of the active players, because I completely agree. It's not just one thing acting in isolation or a vacuum. The ecosystem of the genes making the proteins doing interactions, the proteins regulating those genes, the proteins stopping and starting things and changing how chromosomes are folded. All of that are these multidimensional perspectives that are important to incorporate. So we've been able to build up sort of two of those dimensions, and then there's still several layers to add on to it. Yes, we want to know how that all plays out. That's like a lifetime, but we're getting there. Yes.
Excellent. Online, we're going to try again. Laura Matecha. You are muted for a second.
Thank you.
Yes.
I was wondering if you feel that this protein or this syndrome might have anything to do with like the common breeds they use for service dogs.
Yes.
A lot of the times it's, you know, German Shepherds or Labrador Retrievers. My daughter has a service dog that is not one of those breeds. She actually has a rescue dog that works as a service dog. And I'm just wondering if this genetic mutation might be part of the reason why they use Labrador Retrievers and German Shepherds most often.
Good, really good question, Laura. You've tapped into another arm of my research program that we've been thinking heavily about. So when I think about these mutations and the hypersocial behavior, I think of two applications immediately. One is for shelter dogs, where we have very little information on the dog's history. And what could really be useful is having the genetic information where I could help predict how social of an environment this dog needs to be a successful, happy, healthy dog. The other aspect, like you said, Laura, is that service dog, the assistance dogs. So I've been working with Canine Companions for Independence. We've got data and genotypes on over 1,000 dogs because we are exactly looking at this. The question is more nuanced because we want to take into account all of the different specialized trainings that the dogs can be. Like, are they the most intense of the assistance dogs, or are they more of the casual? And on that spectrum, we're trying to find out, how do these mutations predict the best type of service role a dog can function in. Now, your question did also have the kind of breed. And I think that there's a few standard breeds that these kind of assistance dogs are generally used for, but then there's also like you get a lucky kind of lottery dog that has not the typical breed composition, but is just a fantastic service dog. I don't have a lot of data on those, but I suppose this is a great time to advertise. If anybody wants their dog genotype for this, all you have to do is send me an email.
I definitely will, and I know where your office is.
Yep, send me an email. I'll send you a sample kit. I love this. This is so exciting for me. And I have reports that I generate for anybody who sends me their dog sample. It's at no cost to you because this is something I love doing. And it helps me build my database. So we've got over, I think, 8,000 dogs genotyped, which 8,000 canines. I have a lot of wolves and coyotes. FYI, we never see these mutations in coyotes. Zip. No, nowhere in coyotes. Only if a coyote is a dog, coyote hybrid, do we find it. So yes, email me, Laura. We can see if your daughter's dog is carrying any of these mutations.
How many chromosomes?
Thank you.
Dogs have 38 autosomes and then their XY chromosomes.
Maybe like 19 and me. kind of thing.
It would, well, I don't do ancestry, breed ancestry. I'll only do the hypersocial mutation genotypes.
We actually did the breed ancestry with this dog, with two of the services.
Send that with, I'd be curious to see, because then I could show you other dogs with other mixes that are self-reported. But yeah, that'd be great, Laura. Thanks.
Thank you.
Yeah, thanks for sharing your experience. I mean, it's a fantastic talk. So I'm just wondering that oftentimes with complex phenotypes like social behavior, we think that there is a genetic element, but there is also component of environment.
That's right.
So I was wondering, I mean, for a dog to grow up, they're living in a very controlled environment and friendly environment as opposed to a wolf.
Yes.
So what if you did the reverse, like a dog grew up in a wolf environment,
Right.
would it still grow up that friendly?
Yes. So the wolves that we actually included in this initial discovery are raised, they're human socialized, human raised wolves. And this is something that we have a lot of questions and concerns about to make sure that we're able to account for the early environment in which they've been socialized in. So the 8 wolves that are in this study were all raised by humans, and yet their behaviors, and this has been found by several research groups who include human raised wolves, because you can train an adult wolf to behave like a dog, but the dog will always perform better at the dog-like behaviors. So you can take a wolf so far, and then that kind of ceilings out. So the wolves here were all raised by people, and they still had wolf-like behavior, except for that one, which was a little more interested in people, and also carried a couple more of those mutations. So we have sort of those nice one-to-one correspondences here.
But you were thinking about like dogs in the wild? Is that what you're thinking? Or? Okay.
Mhm.
All genetics are environmental.
No, I mean, so this is explaining one aspect of, is a dog interested in people? And we can predict that they have an increased interest to a certain degree. So the impact on that behavior, we can explain that. It doesn't explain 100%. Environment, genetics, always important, and trying to control for that variability is good. And usually neither will fully explain something, but yes.
Excellent. On the very back?
Yes.
Yes. I'm thinking of life expectation between the two animals. The extended life
The
of the
life
dog
expectancy
versus
between a dog and a wolf.
and a wolf, and the health of the dog.
Yes.
If you can make some kind of comparisons, have studies of the human beings, various cultures across the world. Have you found, with some people have married from one location to across the world with their partner, and have found that they live longer lives. The health of them during their lives is much improved in comparison to what you do with dogs and wolves.
Okay, I'm going to try to unravel this. Let me know if I stray too far. The initial part of the question about longevity, dogs and wolves. So we know wolves generally don't live to an old age and die of natural causes. They're normally living 2 to 4 years. I think the average lifespan in North America of my wolf populations are about 4 to 5 years, and then they die. They die of other wolf attacks, they die of car collisions, they die of being shot by people, and they die of injuries when they try to hunt. So they get kicked by an elk or a bison or something. Wolves can live up to 11 to 14 years in captivity. Dogs, of course, I think all know that generally the lifespan is like 7 to 10-ish, give or take. Now, the quality of life is where I'm getting a little fuzzier about how to respond to that. I think my own interpretation of this is that when we know for dogs, so for me, maybe I can tell this with a personal story. My dog, as a puppy, I took her to one doggy daycare a couple days a week just for the socializing experience. And I know that we're all biased and we all think our dogs are the best dogs. And she came home with some naughty report cards. And they tried to talk to me about, well, do you have an invisifence? Because when dogs come into the new location, she gets like really excited and she nips at them. And like, no, I don't have an invisifence. I don't have a fenced in yard. Like, she's kind of no, she doesn't do this at home. And they tried to give me pointers about having her with, corrections at home. And then I changed doggy daycares. Not because of the naughty report card, but I learned how that daycare interacted with their dogs. And their interactions were, I'm just gonna broadly speaking, throw all the dogs into a room, size matched or temperament matched, throw them into a room, call it good for the day. And then I changed doggy daycares to one where it was set up like a person's home. They had volunteers. I was told the volunteers are on the floor with the dogs all the time. I don't have naughty report cards from that location. She stopped going to them because she's now old and slower, doesn't need all of that hype. But those two sort of facilities with the differences in how they interact with dogs, to me, explains her naughty report card. She is on the slightly higher end of these mutational accounts because, of course, I've sequenced my dog, of course. But so her genetics tell me she should be having these personal interactions as much as possible for her to be a happy, not naughty dog. And to me, that leads into the quality of life. If I know what social environment my dog needs in order to be successful, then I should try to find how to give that to the dog. And whether or not that means the lifespan has a higher quality and value of life, certainly that's going to be my answer. But if I don't get it fully, pick me out of the crowd afterwards and I'll see if I can do a better job.
Great, thanks. By the way, objectively, my dog is the best dog. So. Yes, you.
I had a question.
Yeah.
Does Williams disease make any other differences other than the ones that you shared?
Yes. Are you referring to the people or to the dogs?
Both.
Okay. Yes. Well, my answer is yes for both. For the people, there are some medical concerns. Most of them have to do with the circulatory system and the heart. In the heart, there's a valve that becomes broken, basically, for people with Williams syndrome, and it can cause heart disease, severe heart disease. When we look at dogs with heart disease, it's not as common in dogs as it is in human Williams syndrome, but we have several cases where the same congenital heart disease appears in these dogs, and they do carry these same mutations. So even though I'm not a veterinarian and I don't usually study heart disease, I think this would be a very interesting research question. how much of the medical conditions are also shared by Williams syndrome and dogs. And I wouldn't be surprised if they shared more too.
Hi.
Yes.
A while ago, I happened to have watched an episode of Law and Order SVU,
Yes.
where they did have an actress who played a Williams syndrome
Okay.
person. So this was really fascinating to me as a dog owner. And I guess what I'd like to know is what started you on the path? Because it is really different. It's a handicap
Yeah.
in a human, but obviously in studying this, it's a positive for a
Right.
domesticated animal.
Yeah.
So how did you make that connection that you thought to yourself, well, this is what I need to study in order to know why dogs can be so friendly? And lastly, I have 2 dogs and my golden doodle right in front of me just recently, because I was thinking about your experiment with the sausage. There was a donut in a plastic container, and right in front of me, she defiantly grabbed it and tried to open it up and couldn't, and was very angry when I took it away from her. So anyway, back to the question about
Yeah.
what got you on the path.
Yes, good question. So it was 2 events in my life that got me to those connections. And it wasn't even me making those connections. The first one was the gene itself that I found and published on 12 years ago. I knew was involved in Williams syndrome. And I just thought, okay, well, this is one gene in a list of like 10 that I could go after. And we always kind of caution about that, because I could pick out the favorite story and this would be a favorite story, but that doesn't mean that gene was more important than the next gene or the gene after that. So I shelved it for a while. In fact, I shelved it for like 6 years. It was always in the back of my mind. I'm like, oh, this would be a really good story. And then Princeton was hosting the Animal Behavior Society conference one year. And one of the people speaking at that conference sought me out, emailed me, told me to have coffee with her. Her name is Monique Udell, and she's at Oregon State, and she studies dogs and cats for basically like separation anxiety, attachment, and bond forming between animals and their people. So she pulled me aside over coffee and said, Bridgett, I have found that there are dogs that are centrally in the behavioral spectrum that is described by Williams syndrome. And I said, you have that behavioral data? I said, do you happen to also collect blood samples? So she said, we can do that easily. And that is the start of this story. So she was able to give me an opportunity to think about behavior, and I could help her explain the behavior. So both of our careers have benefited from that. And I think that's why I love this gene complex. That's my favorite gene.
That's great.
Yes, hi. In the fox experiment,
Yes.
they were bred specifically for friendliness, but they were foxes after all.
Yes.
How did that so radically change their appearance?
Their appearance, because what the studies suggest, when we know the progression to go from a wild fox to a domesticated fox, those experiments showed us which traits appeared, which new traits. So what was the first time that a fox had droopy ears? We say, oh, 10 generations after we started the experiment. And then we could say, well, when was the first fox that had this piebald coloring? When did that appear? That was generation 12. So all of these traits that we consider part of the domestication syndrome, we have that mapped out for those foxes being domesticated. Now, what modern scientists have done are these meta-analyses. And basically, I can tell you that nearly every domesticated species, be it a cow, a chicken, a dog, a cat, a hamster, a rabbit, a ferret, a guinea pig, whatever else animal I'm thinking of, follow that same progression. And that's astounding. So that means that there's one central molecular network, one control system that the moment you're breeding an animal for friendliness, or let's say for reduced fear, because a chicken may or may not be considered friendly. Who knows? I don't know anything about chickens, but I know that they're not afraid of people, and hamsters aren't afraid of people, and ferrets. So as we go through that selection, we somehow tap into this molecular network that's all connected, and all of these traits change, and they can change quickly. So that's what the foxes are able to tell us.
Amazing.
Please, I am interested over the result of the experiment of Dr. Belyaev with wild fox. Now the latest generation of fox use as a pet or no, and maybe it's not enough generation, or if yes, where it uses pet.
Yes. Okay. So I have several things to say on this. One, the breeding program in Siberia no longer is ongoing. I think they ran into some funding issues and have not been building their population. What they did over the course of those 50 plus years was end up making basically 45,000 friendly foxes. They had a separate experiment for fearful foxes. So very quickly, we can imagine they both created friendly and fearful foxes. The foxes today, the ones that are sort of left around, there isn't a program. You can adopt them and pay for the shipment of them to the United States. Some of them are already in the United States and you can buy them, but you have to be in a state that permits owning wildlife. And you have to have all of the enclosures for them. Pennsylvania will let you do that, and not New Jersey. I've looked into it. I would love a fox. The thing that we've been able to use the foxes for is that the domesticated friendly foxes carry mutations in the same genes as dogs. Not the same mutations, but the same genes. So the same genes are disrupted. So domestic foxes and domestic dogs have the same outcome through different molecular mutations. We also know about the comparison between the friendly and the fearful foxes. So the research team that focuses on this is out of University of Illinois, Champaign-Urbana, and they do a lot of work, both behavioral and molecular, to really understand how domestication changes everything. But I don't believe they're breeding any more of these foxes.
You've met some of these foxes?
No, I haven't. I was very close to, because they have some foxes over in Southern California. So during my PhD, I was very close to them, but I never actually made it out there, because all of these findings have been since I've been in New Jersey. So I haven't really gone back for that.
Okay.
I want them. I want to hug one.
I know, they sound
Like,
great.
desperately squeeze it tight.
And I encourage folks online, if they have any questions, to raise their hands. I think you're next.
Hi. I wanted to ask for clarification about the nature of the transposons. I was a little bit unclear because I think you said that, like the more mutations that are present,
Yes.
the more social the animal is likely to be.
You see the details I didn't show. So there are some nuances here that are challenging that I glossed over. So when I have collectively, so even if we go back, I guess, whatever. There. So if we go back to something like this, when we see this x-axis, more mutations and more social behavior. Now, the word mutation, I think, easily is interpreted as a new transposon insertion. And that is not how mutation here is defined. Let me just show you a different slide and we can discuss it. So what I didn't make clear is that we've looked at several mutations, And the mutation here just simply means there's a difference between a wolf and a dog. It doesn't mean that the dog has something new. It just means it has something different. So there's 3 genes here that we focused on. And I've given you a little, it's called a gene model. This is what I think the gene looks like. These brown boxes kind of spaced out, but those 4 boxes collectively make up this gene. Here are 3 purple boxes for the other gene, and there are 3 green boxes for this gene. Now, the scope of evolution that I think about is a phylogenetic tree. You didn't know you were going to get this today, did you? So in the scheme of things, when we think about the phylogenetic relationships, we know that wolves are ancestral and they have lineages of wolf populations that are not social. And then at some point, dogs are domesticated, and let's just say they're all very social. And we have all these breeds, and they're dogs, and they're social. So the mutation in time phylogenetically happened here. We can call this, the being non-social is ancestral. And then those that are social are derived. But that doesn't mean a transposon insertion is always derived. For one of my genes, actually 2 of my genes, we see that there's a normal gene sequence in the wolf, and the dog has the transposon insertion. I think this is that intuitive interpretation of what a mutation means. But for the gene that I talked about today, the one today, the transposon is ancestral. Wolves have this, but because that transposon makes them non-social in a simplified way. And the loss of this transposon is what makes this dog hypersocial. But the mutation is that it doesn't have the transposon. This is the derived allele. It is the new version of the allele. So when we go back to that graph of the more mutations, then we're looking at whatever that allele is composed of, transposon insertion or no transposon insertion. Those are both the mutations that happen here. Does that help? Well, I'm so glad you asked that question. That was very keen of you.
Great. Corner in the back.
Yeah, I had a question. I was going to ask you about the other subgroup in Siberia, the wolves that were bred to be like more aggressive and fearful, but you already
The
talked about that.
foxes, they're foxes.
Yeah. Yeah, the foxes. That's what I meant to say. Yeah, I just like this research is about like what makes the wolves and the people with Williams syndrome like sweet and friendly. But I was wondering about like the opposite end of that. Like how much do you know about that? If there's like a group of people that maybe have more in common with the wolves,
Yeah.
like maybe like like psychopaths.
Yes.
Basically, if you know anything about that, the
Yes.
genes that cause that, and if they have more in common with like the more
I-
aggressive animals.
Absolutely. Really good question. And as someone who normally has never studied behavior, I get reminded all the time by my colleagues that because behavior is so complex and so nuanced, it is not a one or the other option. So being friendly does not exclude also being aggressive, or also being depressed, or also being anxious, or also being happy and elated and everything else. So the spectrum on this is not just, are you aggressive or are you friendly? But we have the friendly component measured. In fact, Williams syndrome often results in people being very emotionally distraught, upset, depressed, because they're so driven for these interactions, and those interactions are not reciprocated to the extent that they're desiring. So it's could be a very sad life, although there are many avenues for which Williams syndrome people can find those needs. For dogs, I mean, I think that's the same thing where I saw my own dog turn into a naughty dog, but that doesn't exclude her having the option of being an aggressive dog overall. I mean, she's not, but that being friendly does not exclude the other behavioral features. So it's not a spectrum of one or the other, the multi-dimensional spectrum.
So I've been holding you up for questions.
No, this is great.
Okay, great.
I'm happy to go as long as you'll let me.
Yes, no, you're welcome.
It's a Saturday. What better thing could I be doing than answering research questions?
Okay.
My husband's like, I guess I'll see you later. Yeah.
Okay. So my friends and I were wondering, there was like the gene with like a lot, or the chromosome with a lot of genes like coding for behavior.
Yeah.
And one of them said it was like more like autism susceptible for the
Yes.
dogs. So I was just curious how it would present itself in dogs, especially like in comparison with humans. And also I know there's like differences with like biological like males and females with how this presents itself. So I was wondering if it's like this
Yeah.
pattern is found in dogs too.
Another really good question. And this is something that, hey, if you want to come intern and you want to explore this in my lab, I would need people to do that. The goal is I don't have, you really told me I could do this. I don't have data measuring autism spectrum types of behaviors, but we do know that these behavioral areas overlap, maybe in inverse directions, but having measurements that are specific to autism spectrum disorders is something I don't have. But I had at the start of this thought, are dogs on that spectrum? Is this something that I should consider? Or is it sort of the other end of processing social interactions? So we do find mutations in that gene, but it's not associated with the behaviors we have measured. I wonder how many different dog breeds or just mixed breeds, how many genetic compositions might have behaviors that are reminiscent of autism spectrum and have mutations in those genes. But again, it's a very complex set of syndromes, and how this gene plays a role in that in dogs, I don't know. But it is always, it's sitting right there. I don't know. Yes.
Hello?
Hi.
Okay. Yes. Hello, my name is Godson Lebrard. I'm a undergrad at Rutgers. Yeah, I had a question about the foxes. So
yeah.
So we see that when you have when we domesticate the dogs, I think I read somewhere that, a lot of scientists think that, dogs are just like subspecies of wolves, like you have Canis lupus and then you have Canis familiaris.
Yeah.
Dogs and wolves, they can breed with each other, and they're interfertile, the wolf dogs.
Yeah.
So I'm here I'm thinking, okay.
I love where this is going.
Yeah, you have the red foxes, Vulpes vulpes, and then they domesticated them with like a lot of generations. Would these domesticated foxes be like a new species, I guess, in a sense, or would they be, the same as like a dog, like what Vulpes familiaris, right? So what was the Soviet finding, Soviet like dudes finding with these?
It's a really good question. So I don't even know how I should start with this, because I could pose you the question: how do you generally define a species? What are the basic things that maybe you would want? And you've mentioned one of them.
Yeah, breeding with each other.
Yes. So the breeding together, this is the biological species concept. It is the most simplistic form of trying to define if two things can breed together, produce offspring, then they belong to the same species. But this is not usually the only definition we use. You've also pointed out that people call dogs either a separate species, Canis familiaris, or a subspecies, Canis lupus familiaris, Canis lupus being of the wolf. I think that, so person to person will have a different answer. My response is, I don't really care what we call it taxonomically. I think I normally refer to it as a separate species because it's so different. And defining a species to me requires several types of data: genetic, behavioral, morphology, ecology, physiology, almost like the domestication syndrome. And on all of those factors, I could tell you if you just gave me those data points, I could tell you if it's a dog or if it's a wolf. And because all of those are so different, I think it's a separate species. And also, it's a human-made species. If we stopped doing what we did for dogs, Would we still have dogs? Would dogs still keep themselves going? We do have village dogs, these dogs that don't have the humans making decisions for them. How many would survive? We usually call those a mud puddle dog because all of the differences between breeds kind of goes away. You have them all mixing together and you get a little brown dog. And that's a village dog. You can have feral dogs, which is totally different. Don't confuse that with a village dog, but nonetheless, you're really onto something. And I might call those experimental foxes Vulpes familiaris, but I don't know. I mean, it's an experimentally made organism, and whether or not it's as prolific as dogs, and that might help also determine the argument of taxonomically classifying it as a separate species. But it's a good question, and no one has answers for that are unanimously agreed upon, how we define species.
Hi, Andy, very nice lecture. I have a question about some people have tigers.
Yes.
Even grizzly bears. This is first generation, so it means that my question is this friendly behavior of the animal, is it induced by human behavior towards the animal? Because there's no time for selection, right? They take first tiger or first grizzly bear, and they are very friendly. And also another question, being in Vietnam, I've seen they offer it, the cage with the tiger, and anybody could enter this cage with the guide. So it was the tiger, the guide, and you, and you could spend 10, 15 minutes in this cage. I didn't try. So again, with coming to the first question, would human behavior initiate some genetic changes, inductions of the genes or suppressing of others?
Yeah. So your question is very good at tapping into the scale that it may not be a gene that has mutated. It might be how that gene has been, how it's controlled. And that is what we can think about as being flexible. So let's just say, for example, I spend the first 20 years of my life eating nothing but rice. And then at age 20 to 40, I eat nothing but fish. I have not genetically changed. I haven't genetically changed to the eating of the rice diet, and I won't have genetic changes to my fish diet. What I would have are changes in how my genes are turned on and off in response to that diet. So that flexibility is always there. And I do imagine that if a grizzly bear cub is being raised by people, there are going to be these genetic expression changes that might help buffer this totally different life. It's not a cub being raised in the wild, but now it has people doting over it, feeding it, whatever, playing with it. It's a different lifestyle. And those changes are probably going to help, but it doesn't. So I just have this overall fear that you can put a wild animal in cages, and for 1 or 2 generations, you might think, hey, you've done it. We've domesticated it. But domestication and socializing and like desensitizing are all very different things, as you've noted about evolutionary selection versus just, can I train this animal to not eat me, kill me, bite me? And I just have a very healthy fear of not going into any cage like that. I don't have the trust that the training will override a genetic impulse. And thanks to YouTube, we have all the videos of when those things go wrong. And even wildlife animals, and I just would much rather not interact, not cross paths with them close up. So there are these changes that could help buffer it, but if that's for a long enough time, so if that grizzly bear cub grows up and is still friendly and mates and has another friendly grizzly bear cub over several generations, you could probably get them to be more on the spectrum of domesticated, but I think that there's a long way to go. Carnivores are difficult. We still don't know why dogs have been like the only sort of domesticated carnivore. Cats, we can argue, domesticated. They perform functions, amazing. Are they like dogs? Some cats are, more cats aren't. So I think that even for carnivores, it's very difficult to know when can you override that system.
Okay, we gotta, let's do a couple more questions.
I
There's back-
got a mic here.
Me? Oh, okay. This is a wonderful talk, Professor vonHoldt. And thank you for your generosity in answering all these questions.
This is my hobby.
I came here because I love dogs. So, but I am also really interested in the translational medicine piece of it.
Yeah.
And I think some of the other people asked about, there are, disorders of social disorders, autism spectrum being one of them. What might your research mean for medicine?
So this is, I completely agree. And this is kind of where I turn it around, because I think what I have leveraged, so maybe I can step back. When I started in the canine research field, I was all conservation, wild wolf populations, I'm saving wolves. And as I sort of started bleeding into the dog genetic realm, we saw how useful dog breeds, dog genetics are for learning about human conditions. So purebred dogs share hundreds of diseases and conditions and syndromes with people. So it's difficult to study the genetics of a disease in people because of all of our genetic diversity, our sort of cultural expansion, populations are mixed. We would need thousands and thousands of people with the condition and without the condition in order to learn something substantial. Dogs, we've basically already done that. Every breed is a genetic isolate. It's like these inbred lines of mice, except there are dogs. And all we have to do are study the genes of dogs with asthma and without asthma, and those dogs are everywhere. And we get clean genetic pictures. So normally what I would say is from our dog genetic research, we could help inform the human medical community about genetic targets, therapeutic targets for humans. In this case, it's the total opposite. The only way I know about this syndrome and these genes are because human researchers have already paved the way. So I think what we can learn is more about how can we serve our dogs better based on what their needs are. I don't think we're going to help at least the Williams Association learn more about the genetics of their condition, because I've actually had to use all of their knowledge to inform me about dogs. But what I think we can learn about is how we interact with dogs. So it's not a biomedical model, but it's like a social model. We know what our dogs will need. We can help give them that. We know what people might need and how dogs interact with people. And that's, I think, the spectrum I rely more heavily on. Yeah, if it was like genetics of osteosarcomas, then our dogs can tell us more than a human study could.
Thank you for the great information. I have one clarification question.
Yeah.
That is, you mentioned that the Williams syndrome is also associated with weakness in heart and lungs.
Yes.
And is that also present in dog?
We've only detected the heart disease in a few dogs. And when I say we've detected, it's that veterinarians have diagnosed this ventricle valve issue and have alerted me and said, I'm sending you all of this dog's blood. Tell me about it in connection to Williams syndrome. I have not seen any veterinary or clinical report of categorizing dogs based on the other dimensions of Williams syndrome. I don't know that people are looking for it. I am in touch with like a dog genetic testing company that gives workshops to veterinarians. And so I've talked to veterinarians about this, the genetics of it and how it can be used. Most of the applications have been for when your dog has to have severe treatment. How does the dog respond to being in a clinic? Are they anxious or not? And help understand what the dog needs to recover, to get treated and recover in a non-anxiety riddled environment.
One more question on that piece. Is it safe to assume that Mother Nature doesn't really like friendly gene? because
Because we don't see this just naturally happening?
yes, because human had to intervene to create dogs. If so,
Yeah.
because it could be one of the reasons is this, these cardiovascular problem and the other issue, but is there other reason why Mother Nature doesn't promote this friendly dog?
I will tell you a brief story about primates, chimpanzees, in fact, that there are populations of chimpanzees in Africa that have essentially what people call, I'm not calling it this, but people call it a self-domestication, where there are populations of chimpanzees that have evolved this very cooperative, friendly, altruistic society. And their neighbors next door do not have that. And there are differences in how those two groups of chimpanzees function. Even bonobos have been brought into this. So in terms of Mother Nature, natural selection has given us examples of where this kind of cooperative, friendly unit can evolve. Now, I just call this natural selection because there are some advantages that these populations might have. They've tapped into it, or evolution has provided them the stage at which these behaviors are better than the other populations of chimpanzees or bonobos. I don't know that it would rise to the extent that we consider them the same as a domestic dog. I don't know that. I don't really, I can't say I've seen the literature on the primates that elevate them to being, these are the domestic dogs of the wild. In so many other species, right, we don't see this. I can say what I do have done a study of is for gray wolves in Yellowstone, to ask questions about not so much friendliness, but the other types of behaviors that have been mentioned, the aggression. So wolves do kill each other. They have territorial battles. They sometimes just have issues that result in the death of the wolf that they're interacting with. And we also think about wolf society as being very hierarchical. And those at the top, the question is, how do they remain at the top? Is it that they beat down the rest of the pack, or is the rest of the pack supporting them? So is it a negative or a positive interaction? And we also have this hypothesis that if you're using fear and aggression to get to the top, then you can't be overly aggressive, because then you're just going killing everybody, even your potential mate. And if you're not aggressive enough, then you're never going to reach the top. And when you're at the top, that's when you can reproduce. So in nature, this is called balancing selection. You have somewhere in the middle that's best. So for wolves, you know, having this option of getting to an extreme case could happen. It happens. Do they live so long? Not usually. Do they have as many pups? I don't know. We see that it's more beneficial to be in the middle. So for the friendliness, I suspect that we're facing very similar hypotheses, that working together, working with others, identifying even cross species, where there's a lot of cooperative and mutualistic interactions. Whether that's driven by friendliness genes, I don't know.
In the selfish gene, there's some examples of this, right?
Yeah, that, I mean, being more of a cultural meme, that the things, and of course, this is a kind of group selection, kin selection. Do you help others in the group? Is it a sort of tit for tat? Are we doing this as a benefit of the group, or am I getting a much more bigger piece of the pie than someone else, but they might think they're getting equal? Yeah, this sort of game theory plays out a lot. Colleagues in my department look at these kinds of scenarios from a purely theoretical realm, that can we see when strategies evolve? What is that strategy? How long can it persist? Does it become the dominant strategy? And they do this in the theoretical realm, but tying it to a species is where I'm far more interested in. And I know that there are cases where we can look at this play out, but you're right, we don't just have these sort of hyper-friendly species walking around, especially with humans on the earth.
So they won't, since they won't be able to come down to talk to you afterward, I'll let the last question come from online.
Okay.
Lucille Joy?
Yes, thank you very much. This is a very informative presentation. I have a more or less comment than a question. Last week on the Nature program on PBS, they showed the first of a two-part series about dogs and the evolution of dogs and wolves. And in the very last part of the segment, it showed urban foxes living and getting their food in the streets of London.
Mhm.
And it was amazing to see the interactions between the people walking the streets in London with these foxes, and the interaction of the foxes within their own population. Is this an example of the I wouldn't exactly call it friendliness, but it certainly is some kind of socialization between these foxes and humans?
Yes, Lucille, I think you're also on to something here. So I think I've got a story for this and then the general comment. The general comment is a lot of research that compares urban and non-urban wildlife is looking for signatures of changes that will help the urban species survive, whether it's change in behavior. We also know they have to have a change of diet tolerance, noise tolerance, space tolerance. So those changes, kind of like the earlier question on bears in cages, a lot of the genes can help mitigate that over a short-term interval. But if those animals are successful and they can mate and reproduce, then we would hypothesize that their offspring will have a little bit more of a benefit than their parents and the next generation and the next generation. So urban living could be kind of the start of an urbanized domestic analog. Now, the other end of that spectrum, my personal or research story, is that we've been thinking about this a lot with coyotes living in urban centers. Coyotes live in the middle of Chicago, the middle of Los Angeles, the middle of New York, everywhere. They're everywhere. And living in the middle of these places, I mean, are you surprised to know that coyotes live in Central Park in and around Manhattan? I'm not surprised, but I'm also like, what are they doing? When are they doing the things that they're doing? So we've done a recent genetic study of the coyotes living out on Long Island. And some of our questions were, where do they come from? So genetically, can we place them as coming from like the New England area, or are they coming up from the South? Are they coming up from mid-Atlantic? Where are they coming from? And then how related are they? Are they reproducing and creating family groups on Long Island? And then the third question I really wanted to know is, are they hybridizing with dogs? Because dogs, feral, stray dogs, always out there. Are coyotes doing something and interacting with dogs? And that was the main finding of my work that I thought was important, because we found that most of these coyotes on Long Island are in fact true coyote. There's nothing but coyote genetics in them, except one pack. One pack of coyote were living on a side of baseball diamond, and they were interacting with people regularly. They were being fed hot dogs, and they would come looking for hot dogs or whatever other human food. And this became a problem. And like the other story of Romeo, this pack of coyote were euthanized for safety reasons for people. Yeah. And when I had all of their DNA, and what I found is that that pack actually composed of a female coyote who mated with a dog coyote hybrid, a first generation coyote dog. And that they mated and produced 3 pups, all of which that had 25% dog in them. And lo and behold, I wish I actually knew I would talk about this. I had a figure for it. What we found is that the father, the half dog, and the 3 pups all carried these Williams syndrome insertion mutations, the insertion or the non-insertion. They carried the friendly mutations. Like, well, okay. So something like this, I feel, could help us learn and think about how coexistence might play out where we don't have to kill everything, but also this is kind of a problematic case where wildlife is less fearful of people and might seek out those social interactions. But I do think the urban setting, not only because it's a strong selective pressure, but then also the hybridization could transfer friendly genes into the wildlife. Like this is a complex scenario. And it's a really good question, Lucille, or at least a really good comment, that this could be a landscape where evolutionary change is accelerated by multiple ways, multiple drivers.
Thank you.
Well, thank you so much. Thanks for all the questions. Everybody stuck around.